I was so excited when this drug was approved for sickle cell disease.
Not only because it's the first approved CRISPR gene therapy in the US, but more so because it appeared to work quite well in treating or potentially even curing the disease. But potentially even more exciting is this paper describing a more standard kind of drug, a small molecule, with a similar mechanism to Casgevy,
Perhaps offering a future, more affordable, and easier to administer option for patients. This small molecule, like Casgevy, works by inhibiting an inhibitor of fetal hemoglobin, a protein that carries oxygen in blood cells until its job is overtaken by adult hemoglobin when you're no longer a fetus. But if you have sickle cell disease and your adult hemoglobin causes cell sickling, fetal hemoglobin could step in and rectify things if it's reactivated. But finding a small molecule to make that happen is not trivial. The types of proteins that regulate fetal hemoglobin expression are tough to target with traditional drugs. Yet the authors had hope that they might be able to follow in the footsteps of pre-existing drugs that bind a subgroup of transcription factors called zinc fingers and degrade them by recruiting them to the trash can of the cell. But no one knew if there was a zinc finger that regulates fetal hemoglobin. So they basically threw a bunch of molecules similar to those drugs on cells to see which ones might cause degradation of a fetal hemoglobin suppressing zinc finger protein and not have clearly negative effects on the cells otherwise. One compound which worked nicely was shown to degrade a protein called Wiz, and a slightly modified version of this drug was shown to increase fetal hemoglobin levels in monkeys. Now, there are reasons to remain wary that the specific drug would be safe and effective in humans. It targets a protein that binds at thousands of locations in the genome, and it appears to cause gene expression changes beyond the intended one. But this work is certainly promising, and here's to hoping that a small molecule to treat sickle cell is in fact possible.